Okay, welcome back. Hope y'all had a good Thanksgiving. Couple more weeks, finish things up. I am going to just finish up Streptococcus first, and then we'll talk a little bit about the final, what that's going to look like, what we're going to be doing the next couple weeks. And then several of you have been asking me about that. So let me just finish up the Streptococcus first, and then I'll get into talking more about what to expect as we move forward. All right, so if you remember, the Staphylococcus was on last exam, and I'm just kind of finishing up getting everybody in, taking those exams. So we had so much sickness right before break there. I think by after today, I'll have everybody in, so I'll be able to talk a little bit more about the final next time. The mean was 74 before I curved it at all. So I'm still deciding whether to give a little bit of a curve or not. So I'll let you know about that by Thursday. Okay. And then we started Streptococcus. We've gotten most of the way through this. I was kind of finishing up talking about Strep pyogenes. So we talked about some of the infections that Strep pyogenes causes. It's a gram-positive cocci, remember, Streptococcus is just like Staphylococcus. Some kind of superficial skin infections, but some more serious things like necrotizing fasciitis, strep throat, of course. That's probably what Strep pyogenes is best known for causing. We talked about some of the virulence factors. M protein was one of those virulence factors. It's an antiphagocytic factor. The problem is that the antibodies that we make in response to Strep pyogenes sometimes cross-react with that M protein. One complication can be rheumatic fever. We talked about that. Another complication is with our kidneys. So the acute glomerulonephritis obstructs the blood flow in the kidneys, and sometimes that damage can be permanent. Again, this is when you typically, more often, when you have a strep throat infection that is not treated with antibiotics or maybe only partially treated. Even when you do everything right, I have a colleague that used to work here that every time he got strep, he would land in the emergency room because his kidneys would start to shut down. All right, so you give antibiotics to kill the Strep pyogenes, but really there's nothing that you can do for that autoimmune reaction that can happen other than giving immunosuppressive therapy and try to dialysis if necessary in extreme cases. All right, so moving on, I'm just going to mention kind of briefly a couple other streps. If you're in lab, hopefully these look a little bit familiar. I'm not going to spend too much time on them. The strep agalactiae is a beta hemolytic strep. Remember, that's describes total red blood cell lysis when we streak that organism out on a blood agar plate. It's considered group B. So there's two different ways that we classify our Streptococcus. One is by hemolysis patterns, whether they're alpha, beta, or gamma. And one is by these Lancefield groups, these antigens on the surface of Streptococci. They're actually sugars, things that typically aren't very antigenic. We realize that we can use antibodies to kind of put our streps into group. So that group B, Lancefield group B. The strep agalactiae forms capsules. Let's see, what else was I going to say about that? And the capsule is actually a T-dependent antigen. All right, so you can't forget all that stuff that we just talked about with immunology. Remember, a sorry, T independent antigen is what I meant to say. Hope you can read my writing. T independent antigen. So that means that the capsule for Strep agalactiae, our B cell receptors will recognize that capsule, and that will activate the B cell without any help from T helper cells. Okay, so we talked about that kind of response. You only get IgM antibodies, and you don't get any memory from that. It's not as good a response. What's the reason that I bring that up is that, remember, T-independent antigens, young children don't have very mature B cells, so they don't have a very good T-independent antigen response. Okay, so remember, young children have a poor response. So they're especially susceptible to these kind of antigens. So Strep agalactiae typically is not going to cause infection in adults. It's typically part of our normal microbiota, maybe vaginally and other places as well. But pregnant women need to be tested for group B strep, because if they're positive, I think they're tested end of second trimester, beginning of third trimester. And if they're found to be positive, they're given antibiotics to try to get rid of it, just so babies don't pick up the Strep agalactiae and get some really severe infections. So meningitis, for example, in a newborn baby, really severe, but totally preventable with good prenatal care. Okay. And then the last strep that I just want to mention a little bit, I'm not going to talk about virulence factors here. There are some specific ones, but the alpha hemolytic streptococci, Streptococcus pneumoniae. Of course, the other one is Streptococcus mitis that you, again, if you're in lab, that should look familiar. The mitis is mostly just kind of tooth decay, makes capsules, sticks to our teeth. Strep pneumonia is a lot more serious, the disease that we see there. And this organism will often present as a diplococci. So it doesn't really form those big long chains like some of the other Streptococcus. Really three major kinds of disease. Well, one's not quite as serious as the other two. Pneumonia, you get that from the name, right? This is the most common cause of bacterial pneumonia. It's diagnosed often just by listening to breathing sounds. So some kind of crackling that you can hear when the patient breathes. That's due to the inflammation in the lungs, edema in the lungs. Patients often experience some cyanosis, which means basically they're not quite getting enough blood. Their skin might turn a little bluish, fingertips, especially extremities. It is very treatable. With all the Streptococcus, we really don't have very much bacterial resistance to antibiotics. It's probably like one of the only organisms that I would say that about, but we don't see a lot of resistance. So it's a very treatable kind of pneumonia. They will typically give a couple different antibiotics that work different. Each of them has a different mechanism of action, just kind of make sure that you knock it out, because it is serious pneumonia, but again, it is very treatable once we get on it. Strep pneumonia can also cause ear infections. Otitis media, especially common in young children because their ear canal is more narrow, it doesn't drain as well, often can follow some kind of viral infection. And also meningitis. So this is one of the organisms that can cross the blood-brain barrier, cause inflammation in the meninges, which is the covering of the spinal cord and the brain. Really pretty high mortality from that, and the people that survive are often left with some kind of permanent damage like mental retardation, blindness, deafness. So that's a bad one. It's less common now, though. Thankfully, we have a pneumococcal vaccine. So this organism used to be called pneumococcus instead of Streptococcus pneumonia. And now we have this pneumococcal vaccine. The trade name is Prevnar 13. So you might have heard commercials for that one. Basically, they've taken 13 different, there are 13 different strains well, there's more strains than that, but they tried to take the 13 most common strains, variations of the capsule that's produced by strep pneumonia, strains of the capsule. And remember, capsules are typically polysaccharides, so they're typically sugar. We talked about how usually that's not terribly antigenic. Typically, our immune system doesn't notice that very well. And that's the case here too. So we've conjugated it with a antigen from the bacteria that causes diphtheria. All right, so we see some similarities to the malaria vaccine, where we took something from that Plasmodium, the protozoa that causes malaria, and kind of recombined it with something that is more antigenic, so our immune system notices it. So combined with, I think it's a surface protein from the organism that causes, oh, diphtheria is really hard to spell. I think it's diphtheria. PTH, diphtheria, okay. So now this is a vaccine, I think it's given in 4 doses to babies. So Prevnar 13, you give it both to babies and to older adults, I think over 65, once they're more kind of susceptible to getting pneumonia. And it's been very effective. I didn't post this chart, but I got this, I think I got it from the CDC. Yeah, I got it from the CDC. So when we first started giving this vaccine, late 90s, it was a little bit different. We only had 7 different strains that we mixed in there, but right away, almost cut in half the incidence of invasive pneumococcal disease. So that means when you have strep pneumonia in a place in your body that is normally sterile. So whether it's your meninges or alveoli of your lungs or in your blood. And then when we kind of made the vaccine a little bit better, included some more strains to it, cut it down even further. All right, so this is not only cutting down on invasive pneumococcal disease, but also ear infections, which is still somewhat of a problem. I hear about it less and less with young children, just having ear infection after ear infection after ear infection. So it's been particularly helpful there. All right. Questions about Streptococcus, comments? We won't get into like the antibiotics they're used to treat or anything like that. Okay, so let's talk a little bit about what the final will look like. All right, so first of all, clear your schedules for 2 weeks from Tuesday, not next Tuesday, the week after, right? So, and also try not to get sick. I know that's a tall order. Anticipate that we'll have to give some makeups, but really we have till the end of finals week, right? And at that point, if I can't get you in, I will have to give you an incomplete. So you'll have to come at the beginning of next semester. So do what you can to mitigate your risk of getting sick. I do not want to be doing this online. I haven't decided for sure whether I'm going to be willing to do that, but it's a lot of work to get my exam into an online form. So I'm probably leaning on the side of you know, giving you an incomplete if you can't make it. So try to be there. I'm usually, you know, people that have, you know, 3 exams on one day or whatever, I'm willing to reschedule, but think about whether you want to ask your smaller class to reschedule your exam rather than mine. It's been tough just getting like 15 or 20 people to reschedule from the last one. So, so what it's going to look like. So I would suggest studying especially the stuff that we're doing this week and next week. So our pathogens, and we'll talk more about that. That's going to almost as many questions as a regular exam, but you know, maybe 10 or 15 less questions than a regular exam. So we're going to hit that pretty hard. That's, approximately a third of the exam, maybe just a little bit more than that. And then the portion of the exam that's cumulative, the smaller portion is multiple choice. All right, so we'll talk specifically about that in a minute. And then there'll be a matching of terms part, which is a little bit more. This is the part that students do well on. It's just, again, it's terminology, it's vocabulary. Okay, so I've made that kind of the biggest portion. And again, we'll talk about each of these three in particular in more depth. All right, so these are the pathogens that I am planning right now to cover that I'm thinking that we will probably have time for. So we've already done Streptococcus. I think right now that we may be able to get through all of these. If I have to knock off Bordetella, that would be the one, whooping cough, that I would probably not do. But I think we should wait and see about that one. The rest of these that I think we'll get to. So mostly viruses next week, try to get through the bacteria this week. There's a couple on your learning objectives, there it is, that we are not going to get to. So I'll post a kind of more up-to-date learning objectives with the ones crossed off that we're not going to get to. So there's a few things like tetanus on there that I couldn't see that far in the future when I printed that at the beginning of the semester. Okay, so of these pathogens and these infections, what are you going to want to focus on? Morphology. So Streptococcus, for instance, that's a gram-positive cocci. All right, Neisseria, that's a gram-negative cocci. So just kind of the basics about the organism. I did post this, by the way, as well as with your PowerPoints. Disease manifestations. So anything in particular, like, if you saw some particular symptom, would that give you a big tip off that it's, bacterial meningitis or whatever? Virulence factors is probably the one that's going to take a little bit more repetition and review, because that's going to be a little bit less familiar probably, but that gives us a chance often to go back and kind of tie in other things that we've talked out through the semester. So back to cell structures and tying in stuff from immunology that we just did. Treatment, we'll probably spend a little less time there. In some cases, I've dropped to that portion just so we don't have to go back through and like memorize antibiotics again. Is there a vaccine? If there is, you should know the name of it and how it works, what kind of vaccine it is. All right, so we kind of talk about a lot of interesting stuff. We might talk about how it's transmitted as well. That would be another thing to include here. In fact, let me just write that in. That's kind of an important thing. Hopefully, once you know a little bit about it, that will be a little bit more intuitive. And then the cumulative material, this is the thing that I know is a little bit more worrisome. We've had a lot of content all semester. I will say that I'm not going to ask you any of the history back from the very first day of lecture. And chapter 12, that was the eukaryotic chapter where we talked about protozoa and fungus, algae just for like a second. I won't get into that terminology that we had with the fungal structures, Hostoria or mycelium or mycorrhizae. Don't worry about any of those fungal structures. There was really only a couple of terms we had with protozoa. Takeaway from that chapter 12 is just, do you know the difference between a plant and a fungus and a protozoa? So maybe some differences in the cell wall, or, has peptidoglycan, what doesn't? So just kind of also, between archaea, bacteria, just a little taxonomy, the difference, kind of the main difference between those things. That's mostly what you want to take away from chapter 12. I'm not going to ask you anything about specific antibiotics, so you don't have to go back through and remember what rifampicin does, for instance, or polymyxin. We did only about 4 or so classes of antimicrobial chemicals. I've cut that way back from years past, but I won't ask you to go through those chemicals or anything about those. So that kind of cuts down a lot of what we did back in exam 3. But I am planning on, I don't have the exam totally tweaked and fine-tuned yet. But you're going to want to, if you do go back and review a little bit from the semester, especially pay attention to chapter 3. That was our cellular structures, more so the prokaryotic ones, not less so the eukaryotic ones. Chapter 5, metabolism, how organisms get energy, how they reproduce. Chapter 7, the genetics, and then hopefully the immunology stuff. Whoops, over here is fresh in your mind. Also, chapter 14, that was infection. So to me, those are kind of the main idea things from the semester. So there's, like I said, there's only going to be about 30 questions. 10 of those questions are actually assessment questions that everybody in the course that's teaching right now is asking those same 10 questions. So they're questions that we agreed on like a number of years ago, and we ask those every semester. So some of them I did not write, but I had to include them on there. And I think they're all reasonable questions. So that's the multiple choice. Again, I would maybe spend a little time just kind of flipping back through stuff a little bit, trying to familiarize yourself again. Spend more time on the new stuff that we're doing, just because there's less of it and there's going to be more questions that you know on that. The terms. So this is what the matching is going to look like. Again, if you do go back and study cumulative stuff, I would pay particular attention to vocabulary. I've tried to bold things in your learning objectives that I know are kind of vocabulary words. So start there maybe. But this is what the matching is going to look like. There'll be either 12 or 15 terms on a page. And then I think on the last page, I have the same number of descriptors, and all the other pages, there's one extra descriptor. So obviously, whenever you're doing matching, you're going to want to, let's see. So once you figure one out, Emden-Meyerhoff, that's the same thing as glycolysis. You know, once you figure it out, you're going to want to cross it out. Students that took this last year, I had to give it last year online, and they were at a big disadvantage because when you're taking it online, you can't. Yes, I used that one already. All right, and just from my example here, you can see that some of these terms have nothing to do with each other. So, for example, coccus, right? Round cells. You're not going to get that mixed up, hopefully, with a facultative anaerobe. Like, you really just need to know kind of the main idea of what that is. In some cases, there's two closely related things like lag phase and log phase. Right, that makes sense that if you're going to know what a lag phase is, that you would know how that's different from the log phase, how we describe bacterial growth, or cell-mediated immunity and humoral immunity. Know the difference between those two things. So that's just an example there. All right, like I said, I'm still kind of tweaking and getting rid of stuff that I didn't like from before, and putting something in that seems more reasonable. So, all right, any questions? All right, it is longer. We have an hour and 50 minutes to take the exam. I would say you are used to having way more time than what you need. And this is a little bit more, if you've been using an hour and 15 minutes to take their exam, our other exams, you're going to be a little bit more time crunched for this exam. But again, it's longer, but I think it's very reasonable. All right, so be thinking about that. Ask questions as you think of them. All right, so let's move on. So we're still in chapter 19. Chapter 19 started with the gram-positive cocci, the staph and the strep. Now we're going to move into the gram-positive bacilli, or rod-shaped organisms. Okay. we used Bacillus, maybe you recognize that. We used that in lab a little bit this semester. When the genus Bacillus grows up on an agar plate, you get these really wrinkly colonies with this really irregular edge. So it's kind of easy to recognize if you've got some maybe Bacillus contaminating another culture. This is a genera of organisms that mostly they're environmentals. So that means they're living in the water and the soil, basically outside. They are saprobes. So that's a term that we use to describe, we said lots of fungi are saprophytic. Remember, that means that they live on dead or decaying organic matter. All right, so dead or decaying organic matter, saprophytic. So that describes most of them. There's a ton of different species in this Bacillus genera, at least 50. One source says there's like 200 of them. In general, they're aerobes. All right, so that distinguishes them from another closely related genus, the Clostridium, which we'll get to next. Some of them even secrete antibiotics. Bacitracin, for example, comes from one of the Bacillus organisms. One thing really special about them is that they are able to form endospores. So remember, endospores are a survival mechanism. They can kind of hunker down in this endospore state and just, basically not be doing any metabolism, especially if the conditions are getting particularly harsh, there's not enough nutrients around, or it's too dry, for instance, then they are able to sporulate. Okay. Note that they sporulate in the environment. Okay, so they're not going to form spores when they're in the host. There's only a couple examples of Bacillus that do cause disease in humans. They don't make spores when they're in us. They're gonna, again, make spores in the environment. If they make a capsule, they would do that in the host and not in the environment. So I'll just make a note of that. Form capsules in the host. Okay, so remember, functions of most organisms make some kind of capsule that keeps them from getting phagocytized easily, keeps them from drying out, helps them adhere to surfaces. So they will do that when they're in the host, but then not in the environment. Okay, so just a little review as some of you are thinking about taking a lab final next week. When we spore stain a Bacillus, Schaefer-Fulton spore stain method, we use a malachite green. We used a really concentrated malachite green and let it sit on those cells for quite a while. The malachite green penetrates into the spores. So the spores here are going to look green, and then the vegetative cells, the metabolically active cells, are going to take up the saffredin that look kind of red. All right. Okay, so some examples of bacilli. Bacillus subtilis is the one that we often use in lab. You probably have some on the bottom of your shoe right now. It's ubiquitous, it's everywhere. The pathogenic ones, B. cereus, that causes food poisoning. And that, again, it's in the soil, it can be in dust. So if your food gets contaminated with some dust that has some B. cereus in it, and it is allowed to grow up, it's in a nice, warm place, and it grows up and makes spores, then when we ingest the spores, that can get us ill. It's not a very serious kind of food poisoning. The organism that we're going to spend a little bit more time here on is Bacillus anthracis. That's the causative agent of anthrax. Okay. All right. So let's look at a few virulence factors before and epidemiology before we think about how anthrax kind of presents itself in what kinds of disease. Soil reservoir. So those spores are in the soil, and so people will get anthrax when they ingest those spores. Here's another. Here's a stain of Bacillus anthracis. I'm not sure if this is gram stained actually, or if it's just kind of somebody put some crystal violet or something on these cells, because the spores here aren't really taking up any dye. They're kind of clear. But Bacillus anthracis has a very characteristic kind of boxy shape. That's kind of unique. So you can, it's one of the organisms that you can kind of, do a stain of and probably identify it just from a stain. They tend to form chains. We consider this a zoonotic infection. So that means even though the reservoir is typically in the soil, spores are in the soil, animals will ingest those spores and get sick, and then we typically would get sick from animals. Okay, so we'll say spread between people and animals. Okay, so people only get anthrax when they're in close contact with animals that are infected. Of course, the big exception to that, and one of the reasons that I like to talk about anthrax, is the threats of bioterrorism, which, because the organism can be prepared into spores, it can be dispersed rather easily. You don't have to have a live organism dispersed, you can just disperse the spores. So terrorist attacks back 9/11, 5 people died of anthrax when those spores were sent in the mail to Congress. So humans, though, don't pass it to humans. I suppose it could be if that was done really intentionally on purpose, but typically that's not going to happen. Okay, so virulence factors. So organism, like I mentioned, it does make a capsule. Remember, in the host. Okay, and it's a kind of a unique kind of capsule. It's primarily made of poly-D-glutamic acid, which is actually a protein. So we said, usually capsules are polysaccharide in nature. Remember when we do a capsule stain, again, reviewing for those of you in lab, typically we stain the background because the capsules themselves are hard to get dye to hold on to it. So we stain the background. In lab, we used Congo red, if you remember. And I don't know what kind of capsule stain they've done here, but you can really kind of see this almost halo around the bacilli there. Even though the background's not stained too darkly, they've stained the cell walls and the background so that you can kind of see that capsule. Okay, so the capsule there does what capsules do. They block phagocytosis, and it also kind of resists complement. So like that C3b fragment that would bind to bacterial cells, it's not able to do that. Okay, so it's not going to be our macrophages, for example, are not going to be as efficient at phagocytizing that organism. Exotoxins, there's actually 3 of them, but the third exotoxin acts kind of just as a delivery vessel for the other 2. So sometimes they call it a Trojan horse. It just gets the other 2 toxins inside the cell. And the name of the toxin leaves no doubt as to what they do. Edema toxin, that one causes swelling in tissues. Okay, so lots and lots of inflammation, uncontrolled inflammation, and lethal toxin causes cell death. All right, tissue necrosis, in other words. So anytime necrosis just means death of cells, death of tissue. Anytime you see a blackening of tissue, we would call that a kind of necrosis, and we'll see that in a minute. All right, so that lethal toxin basically alters a cell signaling pathway in the cells and leads to apoptosis. All right, so there's 3 different ways that we can get anthrax, and it's which kind of anthrax we get depends on how the host is exposed to the spores. Okay, so really you could consider the spore formation in Bacillus anthracis as a virulence factor, because that is how that's basically how we get sick. We don't get sick when we're just exposed to the vegetative cell. All right, so the cutaneous anthrax, that's when the spores are going to enter through a break in the skin. Okay, and this is the most common form of anthrax, and it's also the least severe type. So it's least likely to kill you if you get cutaneous anthrax. Very commonly, you might have, small little cuts around your cuticle. So that's a common place that we see the manifestation of those anthrax spores. Okay, so the spores germinate in our body when they enter through small cuts and form a nodule. A nodule, that just means it's kind of a abnormal aggregate of cells. And that can slowly start to cause the death of tissues. So eventually that nodule will turn into an eschar. Okay, that just means a black scab. Okay, so eschar formation, that's supposed to be, it looks painful, but that apparently is not painful. We see some other symptoms in the host, like fever, for example, malaise, headache. But the scab itself is not supposed to be painful. And most of the time, that actually will heal up and scar, and it will be fine. Occasionally, the toxins that are produced there can go systemic, meaning you can get your bloodstream. And when that happens, about 20% of the time, it would be fatal without treatment. Okay, so even if you do nothing, you're probably going to be just fine. But it does have the potential to go systemic. All right, who's at risk? It's really only people that work in close association with animals. So, and it's very uncommon in the US. I think we have, like less than 20 cases of this a year in the US, because our animals are pretty healthy. All right. There are some areas of the world where the spores are pretty common in the soil, like in the Middle East. Not so much in the US. In areas of the US where the spores are more common, they probably routinely vaccinate their livestock. I think in North Dakota, they do that. I believe around here, they don't routinely vaccinate for anthrax, but if they have one in their herd that gets it, then they do. Okay, so it's very not common around here. So again, only if you're traveling are you likely to be exposed to that. The second way we can get anthrax is if we inhale the endospores. So that's considered pulmonary or inhalation anthrax. It's also known as wool sorter's disease. So again, people that are in contact with animals, think about shearing sheep. I've never actually really seen a sheep sheared, but apparently it can be kind of a really dusty thing. You've got a bunch of this going on at once. If the sheep has anthrax and, you know, breathing it out, those spores can be inhaled by us. So again, also known as wool sorter's disease. That kind of reminds us, again, people in association with animals. This is the deadly kind of anthrax, almost 100% if you don't treat it. Typically, what does the disease look like? It often will kind of manifest in like 2 different phases. So remember, you have a incubation period, which I believe is fairly short for this, like hours, maybe days. I don't actually have that information for this. And then, but incubation, no symptoms. Remember, prodromal period of an infection is when you have mild symptoms. And for anthrax, they are kind of like flu-like symptoms. So things like maybe a little headache, maybe a little fever, maybe a little, you know, tightness in your chest. And then sometimes patients kind of feel better for a day or two. Those early prodromal symptoms will resolve. And then kind of bam, they hit the invasive phase. Another name, I think we called it the illness phase before, where they spike a really high fever, uncontrolled inflammation from that edema toxin, pulmonary edema. And if you look at a chest x-ray of somebody that has anthrax, you get this mediastinal widening. So basically, this is wider, that white portion is wider than it should be. And from all the fluid that's accumulating in the lungs. So much inflammation that the blood vessels get leaky, blood pressure goes way down, not getting enough oxygen to the organs, patient can go into shock. That lethal toxin is especially killing macrophages. So macrophages are moving throughout the body, dying. Again, almost 100% fatal without treatment. Once this invasive phase starts, it's pretty fatal even with treatment. I think 50% even with treatment once those kind of more severe, once the toxins really start kicking in and you get severe symptoms. All right, so you can kill the vegetative cells with antibiotics, but then the toxins are still circulating, and that's why it's so fatal. There is a third way you can get anthrax, and that's by ingesting the spores. So gastrointestinal anthrax, this is the way animals are going to get it. They're going to ingest the spores, they're eating off the ground. We can get it if we eat animal meat that has anthrax. All right, so, you know, this probably happened, you know, more so 100 years ago when the farmer says, oh, my cow's sick, I better quick slaughter it so I still get some money out of it. And then people could get sick. All right, so a little different manifestation of symptoms. You get hemorrhaging of the intestines, necrosis, you know, dead tissues, severe ascites. That just means swelling, abdominal swelling. Again, from the edema toxin there, just as fatal as the pulmonary anthrax, so just as bad. How do we treat this? I decided not that probably the antibiotics that I had written down, maybe that's changed over time. You're going to give antibiotics. Let's put a parenthesis here. If someone is suspected to have maybe been exposed to spores, they're going to be given antibiotics prophylactically to keep them from getting antibiotics, or from keeping them from getting anthrax. I think of the 11 people around 9/11 that got anthrax, I think 6 out of 11 of them survived. I think 5 people were killed. So once people started getting sick and they started to figure out what it was, then they gave antibiotics prophylactically to a lot of people, probably saved some lives. In 2012, FDA approved a monoclonal antibody treatment to neutralize the toxins. So if we can figure out that a person is sick with anthrax, right, it's not something we see every day, then we could give hopefully those monoclonal antibodies, and we hopefully would be a little bit better at treating it today. We do have a really good vaccine in animals, and I don't even know, I don't have a name for it there. I guess that's something that I could look up. So it works really good in animals. The vaccine that we have for humans is not as good. You have to give it in, I think, 5 doses over a period of 18 months. And there's a little higher incidence of side effects, and some side effects that are a little bit more serious, like some neurological things. So we obviously are not to the point where we're so worried about bioterrorism that we're vaccinating everybody from anthrax. I think people in the military that are going to be stationed in the Middle East, they have to get the anthrax vaccine, or people that maybe work with animals or work in a lab working with anthrax, then they would get it. But again, and they're not really sure how long it works, so that you have to get a booster every year. So it's not a great vaccine in humans. All right, questions, anything about anthrax? All right, so related organism to Bacillus are the Clostridium. Okay. Also endospore producers. So kind of a related genera, Bacillus and Clostridium, those are the famous spore formers. The Clostridium, though, are anaerobes. Okay, so this organ, some of them are strict obligate anaerobes, some of them can tolerate a little air exposure. But again, if you get sick from some Clostridium, it's almost always from ingesting the spores, because the vegetative cells don't survive in the air very well. Okay, so again, it's more like kind of a virulence factor, ingesting the spores. A lot of the Clostridium, instead of having the spore grow up centrally in the bacilli, will kind of grow up at one end or the other. So you get these kind of, lollipop spores, they might call them, or tennis racket spores. Okay. They are also most often saprobes, so living in soil, sewage, vegetative debris, organic things. In terms of species of clinical interest, a lot of really cool ones, because a lot of them make some really interesting toxins, especially neurotoxins. The Clostridium tetani and Clostridium botulinum, causative agent of botulism and tetanus. Again, it's a toxin that kills you. We are really not going to have time to talk through those this semester. Clostridium perfringens, that's a cause of gas gangrene, and also just kind of a run-of-the-mill food poisoning. So that one is not quite as serious. I decided to take the time to talk about Clostridium difficile, because that's the one that you're more likely maybe to come in contact with a patient that has that than some of you may have already. All right, so we're gonna skip over those. I'll cross out those objectives that I had, those learning objectives for those, and we'll just talk about C. diff. All right, so Clostridium difficile, a lot of us have this organism as part of our normal microbiota in our large intestine, at least in small numbers. You probably have hundreds, if not maybe even thousands of different organisms, different species in your large intestine, and C. diff is just sort of one of the many. Okay, so probably not all of us, but some of us have that as normal microbiota. But it's considered an opportunistic pathogen. So when the conditions are just right, it can take over and start causing a really nasty diarrhea. So what are some of those conditions that would be just right? I got this little table here from the CDC. What are the risk factors? The main one here is being on a course of antibiotics. and probably some kind of broad spectrum antibiotic that's just going to knock out some of the variability in a patient's colon and allow the C. diff to overgrow and kind of take over, start making toxins. Okay, so almost always anybody that gets C. diff, they have just been antibiotics or they're on antibiotics. Being older is a risk factor as well, but I've talked to students that have had C. diff. So it's not just, people in nursing homes, although you are more at risk if that's the case. And again, if you've been in a hospital or a nursing home or are immune compromised or have had C. diff in the past, you're likely to get it again. So let's see. Opportunistic pathogen causes a persistent diarrhea. Oops. That's a hard word to spell. 2 R's. Okay. Try again. Okay, and it can be, at best, it's like a explosive, watery diarrhea. At worst, it can be a bloody diarrhea that actually can, chronic, if it's there for a long time, kind of cause, holes in the large intestine, perforate the bowel even. So how does this get passed? The spores can be spread in the feces, but again, sometimes it's just something that's already there and has the opportunity to proliferate. If you are, working in a hospital and you have to clean up after a patient that's had some C. diff diarrhea, then you've got a gown up, right? All kinds of goggles and everything. That is to protect you, but especially to keep you from passing it on to the patient in the next room. So again, if you don't want to ingest those spores in any way and you don't want to spread it to another patient. All right, so persistent diarrhea. Oh, I found this picture that just looks so painful. That's probably taken from an endoscope. It's a patient's colon that's got a severe form of C. diff, pseudomembranous colitis. All right, so just a ton of inflammation that's causing this ulceration, these kind of membranous, kind of pussy oozingness. This can be a chronic thing. We always start out treating with antibiotics, but sometimes they don't do any good. So patients can have this for months and months, and it's really hard to get rid of. Okay, again, it's the toxins that are causing that. A couple different toxins. One that's an enterotoxin. Remember, not an endotoxin. It's a gram-positive organism, so it doesn't have endotoxin. It's an exotoxin that kind of targets those cells in the large intestine and changes the permeability of them. So they're, more water flushing into the intestine. Toxin B is a cytotoxin, just causing the death of those epithelial cells lining the large intestine. Also, I'll add to that fimbriae. Okay, so the C. diff kind of have to have these structures to stick to the particular kind of cell that they want to in the large intestine, so they don't just get flushed right out of the system. All right, so how do we treat this? Typically, patients are given a course of vancomycin, and sometimes that knocks it out. I forgot to say, C. diff is the one exception to the rule. I said that organisms, Bacillus and Clostridium, don't sporulate in a host. This is the one exception to that. So the C. diff does make endospores, and those do pass out in the diarrhea. Sometimes vancomycin will take care of the infection. We always are going to try that first. There's a ton of resistance to antibiotics with Clostridium difficile, so that doesn't always work. And sometimes we try, course after course of different antibiotics, and it doesn't work. So this is something, C. diff can really contribute to mortality, especially when patients are ill with other things as well. A treatment that has been around for 10 or 15 years is one that will repopulate the large intestine with kind of the normal microbiota again in order to just outcompete that C. diff. And what they're calling this now is FMT, or fecal microbiota transplant. We used to call it fecal transplant, but fecal microbiota transplant sounds a little bit more technical, I guess. So for a patient where the antibiotics aren't working, we do only about 10,000 of these a year in the US, but again, I think it's going to be more popular. You take take a relative of a patient that's willing to donate some feces, and they screen that patient, to, that relative, to make sure that they're, that they're healthy. And then they take a little bit of that feces and with an endoscope would put it in the large intestine of the patient that has C. diff, and that has like a 95, 97% success rate. So it's a really kind of astonishing, simple thing to do. They've been able to put feces in pills now. So when they first started doing the only capsules that would dissolve in the right spot, that would pass through your stomach and not, dissolve until they got to your intestine, were clear. So the first patients that took poop pills, they had clear capsules. So it's kind of an ick factor. They freeze these, so because there's live organisms in there, but you'd have to take 10 of these a day for 3 days. It's a lot. But again, it works. We have them in capsule form now they don't look so nasty. But so they usually take, a patient's relative to do this. But if a patient doesn't have any relatives that are willing or able to donate, sometimes they use universal donors. So some of you probably donate plasma, hey, and make good money from that. Hey, here's another option. So if you're healthy, I think there's only a couple of companies that do this in the US, and I know one of them's in Massachusetts, but if you end up living close to one of those companies, and they screen you and you're healthy, you could make some good money from this, donating feces. So just keep that in mind if you ever strapped for cash. All right. So again, it's still, the FDA still considers this an investigative procedure, so it doesn't have total FDA approval. There was one patient that died a few years ago after having a fecal transplant because he died of a strain of E. coli that was resistant to all antibiotics. So he somehow got that from his donor, and it wasn't one of the things that they screened for at that time. So that was kind of a step back, but I think probably that will become more common. Questions about C. diff? C. diff diarrhea is supposed to be like one of the worst smelly things that you can encounter, I can imagine. All right, let's move on. Still in chapter 19, talk about Mycobacterium. So chapter 19 is supposed to be gram-positive pathogens. Mycobacterium, if you'll remember, we've spoken about these here in the past. These are considered acid-fast organisms. So when we talked about these earlier in the semester and we were talking about cell wall structure, we said, well, there's gram-positive cell walls and gram-negative. And then there's this other category, acid-fast cell walls, and that's the Mycobacterium. Your book includes these in the gram-positive chapter because they don't have an outer membrane. So I guess apparently they stain kind of weakly gram-positive, but most of the times we kind of consider these organisms not gram-positive or gram-negative, but we just call them acid-fast because, well, we'll talk about their cell wall. They have this unique cell wall that has lots of mycolic acid, this lipid in it. Why do we care about Mycobacterium? Well, there's a ton of different species, and most of them are, maybe I shouldn't say most of them are not pathogenic. They're just kind of rare, at least around here. Some of the more famous ones, Mycobacterium leprae, that's a causative agent of leprosy. So here's a patient that's had leprosy. That's an organism that will target the nerve endings, and that organism actually can grow better at a slightly lower body temperature. So it's typically going to be targeting the extremities, nose, ears, extremities get disfigured from this really kind of slow growing infection. Mycobacterium bovis, that's TB in cows. So we can get cow TB when we drink unpasteurized milk. It's a little bit milder form of tuberculosis. Very rare now, at least in the US, because we pasteurize our milk and our cows are very healthy. So we're going to spend some more time here talking about Mycobacterium tuberculosis. It's a causative agent of TB in humans, and that organism does not have an animal reservoir. So that's actually made it a little bit more difficult to study it and to try to figure out a vaccine, because we don't have a good model apparently in animals. At the time that we came out with antibiotics to cure tuberculosis in the late 40s, it was assumed that it was going to be quickly eradicated. The American Lung Association switched from focusing on tuberculosis to talking about, an anti-cigarette campaign. That hasn't happened. We have not eradicated it, and we are not likely to do so anytime in the future. It's estimated somewhere between 1 in 3 and 1 in 4 humans are infected with tuberculosis worldwide. It's a complicated disease. Just because you're infected doesn't mean that you're infectious. So it can be latent, and probably is latent for most of those people. We'll talk about that. But it still kills, almost a couple million people worldwide every year. The incidence in the US is much less. It's more like 10,000 per year in the US. So it's not a huge problem here, but globally, especially sub-Saharan Africa, Indonesia over here, I think this is Myanmar. Again, pretty huge problem in some places worldwide. So, and I didn't post this. I was just, I got this from the CDC as well. The incidence, I'd like to see the incidence back like before we had antibiotics, if we have records there. I didn't find that. Incidence peaked here. This is, the AIDS years, the early 90s. So somebody that had HIV is a lot more likely to succumb to tuberculosis. And it has gone down. The incidence has gone down since then, but we're nowhere near like totally getting rid of it. Okay. So how is this transmitted? Okay, I switched the order around here, so my notes are all mixed up. Okay. Like I said, we're focusing on Mycobacterium tuberculosis here, and humans are the only reservoir. So something where humans are the only reservoir, that's the ones like, smallpox, where we hope that we can eradicate it, because we're just kind of passing it around back and forth. So humans are only reservoir. How is it transmitted? Droplet nuclei. In other words, it's airborne. I think you spelled that with an E. Yeah, airborne. All right. So even if you are further than 6 feet away from someone, it's passed on those really microscopic droplets that we exhale, and they're gonna stay suspended for a long time. So you can get it even if you are not right next to someone. Luckily, it's not very efficient. So most of the time, even when you're exposed, you're not going to get it. It's kind of surprising, because this is a pathogen that's, been around for millennia, but it's not terribly transmissible. What are the risk factors for developing disease? I mean, HIV is a big one, especially if it's untreated HIV, if you're HIV positive and not taking any drugs. This was kind of surprising to me when I looked this up last week. People that have diabetes, why is that the case? People with diabetes are a little bit more likely, especially if their blood sugar is not well controlled, a little bit more likely to have peripheral nerve damage. They have less circulation to their extremities, less white blood cells, our innate immunity getting to different sites in our body. So apparently, if you're diabetic, you're more likely to have severe TB and to come down with it to begin with. Okay. People that are living in congregate settings, so that means people living in jails or nursing homes, homeless shelters, okay, just kind of in close proximity to each other. All right, not a lot of virulence factors to talk about here. Your book only mentions a couple, so we'll stick with those. It gets kind of complicated real quick, and there's a lot that we don't really understand very well. But the acid-fast cell wall composition. And remember, again, for those of you that are in lab and have some reviewing to do, when you do an acid-fast stain, you treat your cells with carbofuscin. Remember, that's a dye that has phenol in it, so it penetrates through that acid-fast cell wall. Remember, again, mycolic acid, it's a lipid. Okay, so that carbofuscin holds on, that acid-fast cell wall holds on to that. You wash the cells in acid dissolved in acetone, and then if it holds fast to that primary dye, we call them acid-fast. Cells that are not acid-fast are decolorized, and then we stain them with methylene blue. So any cells that aren't acid-fast take on kind of a bluish color. This kind of, and I'm way simplifying that cell wall. There's a lot, it has peptidoglycan in it, but it's got a lot of other stuff in there as well. It just makes the cell really resistant. It does get phagocytized by macrophages, but then it can actually live inside that phagolysosome. All those digestive enzymes that are released when the lysosome fuses with the phagosome, oftentimes the organism can survive that. And even in the environment, it's harder to kill. Not as hard to kill as endospores, but harder than just regular gram-positive and gram-negative cells, a little bit more resistant to any kind of disinfectant. Also resistant to many antibiotics because of the decreased permeability. So think about a waxy, lipidy cell wall. Antibiotics just don't penetrate through there. So we have much smaller number of antibiotics that will work against tuberculosis. And the cells grow really slow. I've read that that's because the cell wall just takes a long time to kind of manufacture. Because they grow slow, they also die slow. So patients have to be on antibiotics for months and months and months to kill all the organisms. So really just that cell wall is considered a virulence factor. The only other one that I'll mention here is that cell wall has a glycolipid in it called cord factor. And we don't know exactly what cord factor does, but we know that when we've made Mycobacterium tuberculosis mutants that don't have cord factor, they don't cause disease. So they're not able to, really replicate in the body. We do know that it causes the cells to remain attached. So it's kind of hard to kind of make out the cells in this picture here, but they do tend to kind of clump together. And we think that that's because of this glycolipid cord factor. It probably inhibits the migration of neutrophils, and it's one of the things that it actually keeps the lysosome from fusing with the phagosome sometimes, right? So you can see how that would make it more likely to survive inside the macrophage, right? And there are other mechanisms that we won't talk specifically about that make the phagosome fail to acidify. That's one of the things that contribute to killing by phagocytosis. And remember, macrophages have kind of two levels of activation. They have their normal level, and then they can be kind of activated to kind of work harder and be more efficient at phagocytosis. And they have mechanisms to keep them from doing that. So again, a lot of it is kind of poorly understood, but the cord factor and the acid fast so helps the Mycobacterium just live in the macrophages. That's the cells that they take over. All right, so let's start to talk through the disease. Like I said, it's fairly complicated, and there's things that we don't understand still, but tuberculosis always starts in the lungs, and it can spread to almost every other part of the body, but it always starts in the lungs. So we inhale it, and it targets the macrophages that are in the alveoli. So right at the ends of the bronchial tubes. And that's where the bacilli are going to replicate freely. So when you first, a couple of days after you inhale those, patients get kind of mild flu-like symptoms, not, kind of headache, malaise, that kind of thing. Nothing that really will tell patients that they have something similar. And at this point, some of the macrophages may be able to present some antigen, both by MHC class 1 and MHC class 2. So sometimes we get enough B cells activated and T cells activated that patients clear the infection. But often not. Okay, oftentimes we have to settle for kind of a stalemate with the organism. So patients get these granulomas forming. Granulomas are kind of aggregates of kind of cells and other debris that are kind of abnormal. And we call those granulomas tubercles. Again, and this, we've got a picture over here, represents kind of a stalemate, like I said, between the bacteria and the host, where it's kind of, walling itself off with some dead macrophages and some collagen formation, a structural protein collagen. In the center of the tubercle, though, you've got live bacilli, so live Mycobacterium, but they're just kind of walled off, deep in the alveoli of the lungs. And they can sit like that for years. Okay, so a patient that's got tubercle formation is said to have latent TB. Okay, so, or, primary TB, latent TB. And that's not infectious. So you can have those in your lungs and not even know about it and not be getting other people ill. Not infectious. I'm going to write that there. Sometimes the inside of that tubercle, some kind of like some of those proteins turn and change into this kind of cheese-like appearance. So sometimes we get this caseous necrosis. This picture in your book kind of shows that a little bit. So this is on page 576 of your book. This caseous necrosis, where the inside of the tubercle, again, just best way to describe it, the proteins look kind of oozy and cheese-like. According to, and I'm not really clear if once it turns, the granuloma turns caseous, if that means it's starting to spread and break out of the tubercle, or whether it can sit like that for a long time. Your book makes it sound like, just at some point it can reactivate. My understanding was it gets kind of like that cheese-like consistency when it's getting ready to activate. Either way, once that tubercle is breached, which, when would that happen in the patient? Maybe something changes with their immunological status. Maybe they've got chemo, they're undergoing chemo, or they have some other infection, or they start taking, some kind of drug or something, then it can activate. Okay, and then we would call it secondary TB. Only about 10% of individuals will their latent TB activate. So that's the question. If somebody has primary TB, do you treat it? The treatment process is long and hard. So that's a decision that depends on where somebody lives and so on and so forth. But we think only about 10% of the people will it activate. And now, once those tubercles are breached, now the patient is infectious. And we start seeing symptoms like a productive cough. So, lots of sputum coming up. Sometimes that sputum is bloody, lots of chest pain, and that will progress into, appetite loss, weight loss, chills, sweats, fever. Okay, and even at this point, treatment at this point is usually successful, but not 100% of the time. Okay, so we'll finish up with TB and move into our Neisseria next time. Have a good day.